Good evening, ma'am. Good evening, friends. Today I would like to give an explanation about allostructure of bacteria. I am studying B.Pharm second year in Maharaja College of Pharmacy. So, coming to bacteria, this is the structure of bacteria. Bacteria has a types, and the parts of the bacteria are plasma membrane, cell wall, capsule, ribosomes, pilus, cytoplasm, plasmid, flagellum, nucleoid. So these are parts of the bacteria. So this is the type of a bacterial cell structure. So coming to definition of a bacteria, bacteria are prokaryotic, unicellular, free-living microorganisms which can lead its independent life. They possess both DNA and RNA. DNA and RNA are the genetic material which is present in bacteria. It's a prokaryotic. Prokaryotic means, and these are the unicellular. Unicellular means which has contains only one cellular, free-living. It can live anything. It can live any like, and which can lead its independent life. So it has a genetic materials both DNA and RNA. So before we study the parts of the bacteria are capsule, cell wall, plasma membrane, cytoplasm, ribosomes, mesosomes, nucleoid, plasmid, flagella, fimbriae and pili. So every part has its function, and it's a main function of a bacteria which is moves towards its life. So coming to capsule. So here we see that capsule which is present in the bacteria. The main part of the capsule which is present outside of the bacteria. So coming to the parts, before the study of bacteria is called bacteriology. So coming to the parts and its main function which helpful in the bacteria. Coming to capsule. Capsule is the outermost layer of the bacteria, and it can be consisted into two types. Capsule, which are the main function is the protection for the outer barriers, and also it can be classified in two types, capsulated capsule, and second one is non-capsulated capsule, and another one is non-capsulated bacteria. One is capsulated bacteria, another one is non-capsulated bacteria. So coming to second part, cell wall, which is the undermost layer after capsule and is also used as a barrier. And the main function of the cell wall is it gives shape and rigidity to the bacteria, and it classified into two types according to gram staining. Based on the gram staining, first one is a gram positive bacteria and another one is gram negative bacteria. So coming to plasma membrane, which is the third layer from outside. It is a layer and is used as a barrier to the involving to bacteria for foreign particles. And coming to the fourth part is cytoplasm. Here all organelles are present in the cytoplasm, which is considered in the bacteria. And the fifth part is about ribosomes. Ribosomes are 70S ribosomes type is present in bacteria, and 70S ribosomes are divided into two subunits. One is 50S ribosomes and another one is 30S ribosomes. These are the ribosomes are useful in bacteria are protein synthesis. So coming to next part, mesosomes. These are the intraconvoluted structures of plasma membrane which is helpful in the cell division. So next coming to other part is nucleoid. Here in bacteria we seen that nucleus structure is not that much is present in not well developed in bacteria, and also nuclear membrane is not present, nucleus is not present. So next coming to plasmid. So plasmid. Plasmid is the other part of the bacteria, and they are the small circular extra. So here we see that plasmid structure. You see that these are the small circular extra chromosomal genetic material in bacteria, and they confer resistance to the bacteria. So here two types is there. F plasmid bacteria. Bacteria having F plasmid are called F plus, which means male bacteria, and without F plasmid is called female bacteria. The types of plasmid. So next part is coming to flagella. Flagella are a thin, long, hair-like, slender filamentous arising from the plasmid base. So here you see that flagella which is present from the plasmid and its outer structure which are gone. So coming to flagella, which are helps in the main function is locomotion, to move DNA from one place to another. Flagella is used. Next one is fimbriae and pili. So here you see that pili, which is the outermost structures which are hair-like structures. They are the thick, short, hair-like appendages of the cell wall, which are arising from the cell walls. So that's why the definition is the thick, short, hair-like structures which is arising from the cell wall. So it is made up of a protein called fimbrine or pilin. Pili is two types, pili and sex pili. So the main function of the pili is the transfer of genetic material, input of these genetic material and the attachment, and is also useful for the attachment. So this is the parts involved in bacteria which are useful for the different functions, useful for bacteria which can live to survive in this earth. So these are the parts, and thank you for giving this opportunity to give a seminar on ultrastructure of bacteria. It is the smallest organism which is present in the earth. Thank you very much.